Right, now we are going in detail of oogenesis. Right, what is oogenesis? Oogenesis is formation of mature ovum from oogonia. Is that right? So now we will start that how the oogenesis occur. It occurs in two parts. Oogenesis starts in prenatal life in females even before they take the birth. When female babies are, female fetus, when it is still in the uterus of their mother, at that time their ovaries start working and on their oogenesis process. So in this lecture I have divided oogenesis before birth, that is prenatal development, and oogenesis after birth, which is postnatal development. Now first of all we will talk about oogenesis before birth, that is prenatal development. Actually most primitive, you can say, cells which are going to be germ cells, we call them primordial germ cells. Primordial germ cell first of all appear in yolk sac. Is that right? Around the 4th week. So around 4th week of development, of course in the uterus, in the wall of the yolk sac, primordial germ cell appear, and these primordial germ cells keep on proliferating by mitosis. They are proliferating by mitotic process. Meiosis has not yet started. So these primordial germ cells keep on proliferating and migrating from the wall of the yolk sac towards the gonads of genetic female. This is the gonad, early developing gonads of genetic female, and these primordial germ cells eventually reach to primordial germ cells reach to the gonad of the developing female. Now I have made the ovary of a female and I am going to tell you how these primordial germ cells develop there. Let's suppose this is our first primordial germ cell here. Now primordial germ cell, what is the situation here about the genetic material? Actually it is having again how many chromosomes? If you remember last lecture of meiosis, because this lecture is totally based on the last lecture of meiosis, you have to watch that video before this. Now in primordial germ cell, how many chromosomes are there? One N chromosome, that is 23 paternal, and one N chromosome, 23 maternal. Is that right? So this cell is total having how many chromosomes? Yes, 2 small n, which is equal to 46 chromosomes, that is diploid number. Diploid number, right? These are present in primordial germ cell, and these diploid number of chromosomes, all the chromosomes are single structured, because every chromosome is single structured. So 23 chromosome paternal provide 1n amount of DNA, and 23 maternal chromosome also provide you with 1n DNA. So total amount of DNA is 2n. Is that right? Now when these primordial germ cells are multiplying by the mitotic division, and they are producing the next generation of cells which are called oogonia. So what is happening? Primordial germ cells, via the process of, yes, mitosis, via the process of mitosis, they are producing oogonia. Now these oogonia, because they are produced as a result of mitosis, right? So what really happens? That primordial germ cell before mitotic division replicate their DNA and then divide it equally into 2 daughter cells. So amount of DNA in the oogonia and number of chromosomes in the oogonia is same as in primordial germ cell. So what does it mean? That oogonia, right, they are also having which situation? Yes, 46 chromosomes. They are also like primordial germ cell, they are diploid cells, oogonia, and of course the chromosome amount of DNA is 2n. Is that right? Then usually oogonia initially are surrounded by, you know, cell, epithelial cell, flat epithelial cell derived from the ovary epithelium. epithelium present on the surface of the ovary. Right now, this originally there is one oogonium, and this oogonium keep on multiplying again by mitotic process and keep on producing more oogonium. Is that right? So it means oogonium keep on multiplying by which process? Mitosis. And because they are multiplying by mitosis, so what really happens? That all the oogonia which make more and more oogonia, all of them are having the same genetic material. As you know, in process of division, mitotic division, every cell which is going to mitotic division first double its genetic material and then divide equally into two daughter cell, and genetic material in the daughter cell is same as the parent cell. So again, all the oogonia will have the same 46 chromosomes, diploid number with 2n amount of DNA. Any question here? Now, once they have significantly increased their number, oogonia have significantly increased their number, then a time comes when one of the oogonia start enlarging and other oogonia degenerate, and now eventually you will have one oogonium in a sheath or bag of epithelial cell. Now this single oogonium which is present surrounded by the flat epithelial cells, these cells are called follicular cells. This oogonium enlarges in size and then it converts into primary oocyte. When I say oogonia converts into primary oocyte, I mean that now oogonia are not going under mitotic division once they are transformed into primary oocyte, because primary oocyte will start process of meiosis. Let me repeat it. Look, primordial germ cell repeatedly undergo mitosis, produce lot of oogonia, and oogonia produce lot more oogonia by mitotic process. But eventually, all the oogonia which are produced in big numbers, many of them degenerate. But many of the oogonia enlarge in size and they stop mitotic division. Many oogonia stop their mitotic division, enlarge in their size and get ready for meiosis. These large cells which have stopped mitosis and planning to do meiosis, right? These cells are called primary oocyte. What are these cells? Yes, primary oocyte. Now these primary oocyte, because they are produced as a result of mitosis of oogonia, primary oocyte, they are produced by the mitosis of oogonia, so their genetic makeup is same as oogonia. It means they are also having 46 chromosome. It means they are also diploid cells. Primary oocyte, like primary gamete, also having are diploid, and their DNA is 2n. Any question here? Now, primary oocyte start their first meiotic division, right? But before starting the first meiotic division, in the primary oocyte, DNA replicates itself. If DNA is replicating itself, it means each of the 46 chromosome now convert into duplicated 46 chromosome, or they convert into double structured 46 chromosome. Now, it means when it goes to the next stage, right? What is next stage? Into prophase of the meiosis 1. At that very stage, what is the genetic makeup of this cell, right? Now, in the prophase, genetic makeup of the cell, prophase of the first meiotic division, is that now the cells have replicated their DNA and duplicated their chromosomes. Is that right? But still, chromosomes are 46. But these 46 chromosomes are double structured. What are these? They are double structured. But because they are still counted as 46 chromosomes, so this cell is still considered to be diploid. Right? But because now amount of DNA after replication has become double, this cell in the early prophase is no more 2n amount of DNA. Now the amount of DNA is 4n. That is what is very important to understand, that amount of DNA is now 4n in the primary oocyte, which is entering into first meiotic division. Now during the first meiotic division, at early stage, that is in prophase, these cells, primary oocyte, once they start their first meiotic division, during the prophase, there is a special stage which is called Diplotine stage. What is that stage called? Diplotine stage of prophase. When primary oocyte reach to the diplotine stage of the prophase of first meiotic division, right, they arrest their further progression in meiosis 1. What they have? They have arrested, they stop their further progression in meiosis 1. So what does it mean? That before the birth, all the primary oogonia, before the birth of this female baby, all the primary oogonia have started their first meiotic division. All the primary oogonia have been converted into primary oocyte. After birth, at birth there is no at birth there is no oogonia. What really happens that before birth, all the oogonia, many of the oogonia degenerate, but remaining all surviving oogonia before birth have been converted into primary oocyte. They have been converted, all the oogonia have been converted into primary oocyte. And before birth, all the prime, these are millions. At birth there are 2 million primary oocyte. So at birth, all the primary oocyte which are present in female baby, female newborn infant ovaries, all the primary oocyte have started their first meiotic division, but never complete their first meiotic division before birth, right? And all these primary oocyte which have started their first meiotic division, they arrest their progression in first meiotic division, they stop their progression in first meiotic division at prophase, right? Especially at diplotine stage. Now, so it means female is born with the ovaries with almost 2 million primary oocyte, and of course there is no oogonia at birth, is that right? And after the birth, no further development of, after the birth, because at birth there is no oogonia, so there is no new formation of primary oocyte, is that right? But before birth, lot of oogonia and primary oocyte degenerate, because these cells, germ cells, around the 5th month of intrauterine life, they are about 7 million. But at birth, many of them become degenerated and they are only 2 million. So when female is born, again I am stressing, female baby is born, there are no oogonia in ovary, number 1. There are almost 2 million primary oocyte. All the primary oocyte have started there, guess what? First meiotic division, but arrest at diplotine stage. And all the primary oocyte are surrounded by a flat epithelial cell. You know this, and this, when primary oocyte surrounded by the flat epithelial cell, this is called primordial follicle. Please don't confuse primordial germ cell with the primordial follicle. Primordial germ cell is here. Primordial germ cell is the very early stage of early stem cells from where the ova will develop. Primordial germ cell. So primordial germ cell convert into oogonia. Oogonia surrounded by flat epithelial cell, right? And then oogonia enlarge and convert into primary oocyte, and primary oocyte surrounded by, what is this? Flat epithelial cells. These cells are derived from the surface epithelium of the ovary. These flat epithelial cell along with the primary oocyte, this is called primordial follicle. So what we will say, primordial follicles are having which oocyte? Primary oocyte. Is that right? So when baby is born, female infant is born, in the ovary there are about 2 million primordial follicles, and every primordial follicle has primary oocyte, and every primary oocyte is started at first meiotic division but stuck in which stage? Arrested in diplotine stage. Now after birth what happened? Now it is postnatal development. This is an ovary after the birth. Now after birth, right, up to puberty, primary oocyte remain arrested and most of them degenerate. What happens? After a female infant is born, there are about 2 million primary oocyte along with the epithelial cell. Most of them degenerate, and around puberty, female ovaries, together both ovaries, around the birth they are having about 40,000 primordial follicle. Around puberty, female has how many? 40,000 primordial follicles with primary oocyte arrested in Diplotine stage and not yet completed their first meiotic division. Now, during at puberty, when menstrual cycles or ovarian cycles will start, you must be knowing that follicle stimulating hormone will come and start stimulating the primordial follicles, right? Now, every month in the ovaries, a pool of growing primordial follicles is maintained. This is a growing primordial follicle. It is still having which oocyte? Primary oocyte in Diplotine stage. Now, every monthly cycle, about 5 to 15 primordial follicles start growing. How many? 5 to 15 every month, right? 5 to 15 primordial follicles start growing. Is that right? And then this follicular cells which are around the ovum, primary ovum, they become cuboidal. Originally they were flat cell. They become cuboidal and then they become multi-layered. They become stratified cuboidal epithelial cells. Is that right? This stage of development of follicle is called primary, what is this? Primary ovarian follicle. Primary ovarian follicle. And what was this follicle? In the beginning, this was primordial follicle. What was this? Growing, yes, growing follicle. So every month, 5 to 15 or 20 growing follicle further grow and many of them convert into primary follicle. What are the features of primary follicle? Even though primary follicles still have primary oocyte in Diplotine stage, but it is surrounded by multi-layers of follicular cells. And around these follicular cells, some connective tissue cells also start differentiating from the ovarian stroma. Is that right? But a very important thing which I must mention is that when primary follicle is developing, oocytes secrete glycoproteins outside. Oocytes secrete glycoproteins outside, and these follicular cells secrete same glycoprotein inward. So between the oocyte and the follicular cell, a special glycoprotein layer is present. This is called zona pellucida. This is called, what is this? This is called zona pellucida. Zona pellucida is a zone of a layer of glycoprotein, meshwork of layer of glycoprotein around the ovum, right, outside the ovum and inside the follicular cells, right? And this glycoprotein components are secreted by the ovum outward and follicular cells inward, and this is zona pellucida. So now, what happened with further development of follicle? This was primary follicle with primary oocyte arrested in Diplotine stage. With further development during the monthly cycles, what will happen? That cells will proliferate a lot, follicular cells. Whole follicle will become a large, fluid-filled spaces will appear in between the follicular cells, and they will coalesce or fuse with each other, and this fluid-filled space is called antrum. Is that right? And then some follicular cell remain around the, what is this? Ovum and zona pellucida. These follicular cells which are around the ovum and zona pellucida, these are called cumulus oophorus. Cumulus oophorus. And this is the connective tissue which is specialized. This is a connective tissue derived from the ovarian stroma, and this connective tissue start thickening and make a special covering around the growing follicle, right? This covering is called theca folliculi. What is it called? Theca folliculi. Now this theca folliculi start forming at primary follicle. What is this? Theca folliculi. Now as follicle further grows and it develops a cavity, now it is called secondary follicle. What is it? Secondary follicle. But remember, this secondary follicle still has primary oocyte still rested in Diplotine stage. Is that right? Secondary follicle. It is a growing secondary follicle. This is maturing or growing secondary follicle, right? Which is still having primary oocyte in Diplotine stage arrested, and most of other follicles will degenerate. Usually in the end, only one of the follicle keep on growing. And this theca folliculi which is around it, these cells which are more internally placed, these are called theca interna. And cells which are outside, the tissue which is outside, this is called theca externa. Later on we will learn that theca interna cell along with the follicular cell produce estrogen. That I will discuss in detail in the next lecture, right? Then in next stage, this follicle keep on growing further. But what was this follicle? Yes, this was secondary follicle. Is that right? Again, come back. What was this originally? Primordial follicle with primary oocyte. Then they were growing follicle. Eventually primary follicle with, what is this? Primary oocyte in Diplotine stage. Here is secondary follicle, right? With an antrum, growing secondary follicle with primary oocyte in Diplotine stage. Now, this oocyte will wake up from like a sleeping beauty. It was arrested in sleeping phase. This, what is this? Primary oocyte will come out of its arrested stage, and now it will progress to complete its first meiotic division, right? And about when it will complete its first meiotic division, right, then it will divide into two cells. One cell with very little cytoplasm. One cell will be with very little cytoplasm. This is called first polar body. And other cell will be very large and with lot of cytoplasm. And this is called secondary oocyte. So secondary oocyte is formed by the completion of first meiotic division. Is that right? By the completion of first meiotic division, right? And extrusion of, what is this? First polar body, right? First polar body lies between the zona pellucida and the secondary oocyte. Now, when primary oocyte go to secondary oocyte, right, it means primary oocyte has completed its first meiotic division. And in the end of that, what happens to genetic material of this cell? If you remember the lecture on meiosis, at the end of the first meiotic division, the daughter cells must have 23 what? Double structured chromosome. 23 double structured chromosome. So these cells are haploid as far as chromosome number is concerned. But as far as DNA material is concerned, that is 2n. This is secondary oocyte. Now, secondary oocyte, as soon as it is formed as a result of completion of first meiotic division, as soon as it is formed, it immediately enters into second meiotic division. Is that right? Secondary oocyte enters into what? Second meiotic division. And this fully mature, now this is fully mature secondary follicle. This fully mature secondary follicle is also called Graafian follicle. So what is Graafian follicle? It is fully matured secondary follicle. Is that right? So again go back. We were having originally primordial germ cell with mitotic division, they convert into oogonia. some of the oogonia enlarge and convert into primary oocyte, right? Now they are no more going into mitotic division. Primary oocyte will start their first meiotic division and arrest first meiotic division at Diplotine stage. Of course, this cell will be surrounded by the epithelial, flat epithelial cells, and these are called primordial follicle. All these things occur before birth. When baby is born, ovary has lot of what? Primordial follicles with primary oocyte arrested in Diplotine stage. Up to puberty, all primary oocyte remain in Diplotine stage. Then every monthly cycle, some of the primordial follicles will be activated into growing follicle, and then primary follicle, and then secondary follicle, and then mature secondary follicle will be only one, that is Graafian follicle. Now, what happens that every month when some of the primordial follicles are activated, the primary oocyte in them will complete their first meiotic division, especially in which oocyte? Oocyte which is in the Graafian follicle, right? Complete their first meiotic division and produce secondary oocyte in first polar body. Is that right? Now, as soon as Graafian follicle has what is this? It is around the middle of the cycle, right? 14 days before the next menstrual cycle, Graafian follicle is fully ready for ovulation. And how ovulation occurs? Just before ovulation, luteinizing hormone increases this surge. Concentration of luteinizing hormone in the blood of the female goes up, right? That help it to ovulate. But just before ovulation, just 3 hours before ovulation, how many hours? Just 3 hours before ovulation, secondary oocyte, which was going through second meiotic division and which has arranged its genetic material on spindle in metaphase, right? Now the secondary oocyte again arrest its development where? In second meiotic division at metaphase. I don't know why the ova like to arrest itself at multiple stages, but what happens that every month one of the Graafian follicle which has grown best, usually many follicles start but only one become fully mature, and ovum which is present in that one fully mature Graafian follicle move from primary or complete its first meiotic division, and at the end of first meiotic division there is secondary oocyte and what? First polar body. Then secondary oocyte start its second meiotic division, but it does not complete its second meiotic division. Just 3 hours before the ovulation, it arrests its second meiotic division, right, in metaphase. Is that right? And then this ovum along with some surrounding cumulus oophorus cell that ovulate out, and they are taken up by fallopian tube. Now you can see the secondary oocyte, right, of course with one polar body, which is polar body is usually non-functional cell and eventually degenerate. With polar body, secondary oocyte keeping its what? It's arresting its second meiotic division, meiotic division, arrested at which stage? Metaphase, right? It is arrested at metaphase, chromosomes are arranged here, right? It comes in the fallopian tube. And remaining follicle with some granulosa cells and theca cells that remain behind, right, without any ovum, and they convert into yellow colored mass which is called corpus luteum, which will produce a lot of progesterone in second half of monthly cycle, right? But we'll talk about that in the next lecture, hormonal situation. Right now what I'm concentrating that at the time of ovulation, right, ovum with cell of cumulus oophorus comes out, and cell of cumulus oophorus they rearrange themselves around secondary ovum, and this after the rearrangement these cells look like a crown, so we call it corona radiata, right? This is the ovum, secondary ovum, and it is arrested in which stage? Second meiotic division. At which phase? Metaphase. Now if sperm does not come, Many times sperm does not come. Every female is not sexually active, and even if she is sexually active, she is not getting sperm at that very time all the time. So, if sperm does not come, if there is no sperm coming, there is no magic touch of sperm, what happens? This ovum will wait in fallopian tube from 24 to 48 hours. This ovum, which has started its second meiotic division, the secondary ovum, which has started its second meiotic division, but arrested at metaphase, right, will keep on waiting with great anticipation for the magic touch of sperm. If sperm does not come, this will disintegrate. It will become apoptotic or necrotic. This ovum will die out. Don't tell anyone that females give an egg outside. No, this will just die in the fallopian tube. Is that right? And this organic material will be absorbed by the ovarian, in the fallopian tube epithelium. So it finishes. But let's think of positive things. Let's suppose when secondary ovum along with its crown, corona radiata cells, is waiting here, and happily sperm reach here. As soon as sperm touch during fertilization process, as soon as sperm touch the cell membrane of secondary ovum, suddenly secondary ovum become reactivated. Oh my God, sperm is here and I have not completed second meiotic division. So very rapidly, this sleeping cell stuck into metaphase of second meiotic division suddenly become active, happy. And what it does? It will rapidly complete its second meiotic division, right, and produce one more polar body. And after that, this ovum, right, already there was one polar body previously, now one more polar body is released by the completion of second meiotic division. And this meanwhile, first polar body may divide into two also. You get it? There was first polar body here, it also divide into two and produce two polar bodies, and now one more polar body is produced when secondary ovum divide into definitive ovum, definitive fertilized ovum. And what is this? Another polar body. Now this ovum, which was secondary ovum originally, arrested in metaphase, if sperm does not come, its life is only 24 to 48 hours. But if sperm come and it is fertilizing this egg, so as soon as sperm membrane touch the sperm material touch the secondary ovum, it rapidly completes its second meiotic division, releases one more what? Polar body, and converts itself into what? It converts itself into definitive ovum or mature ovum. This definitive ovum will now have 23 chromosomes which are single structured, single chromatid or single structured, right? So this is haploid cell now. What is it? It is still haploid but now single structure, definitive ovum. But secondary ovum were also haploid but double structured. So definitive ovum was haploid but with double structured chromosomes with 2n DNA material. Now definitive ovum have 23 chromosomes but single structured and 1n amount of DNA. Now this convert into fertilized ovum, right? Now what happens? Its 23 chromosomes are in female pronucleus, and from the sperm this nucleus enter in and it is called now male pronucleus. Now 23 chromosomes are in female pronucleus and 23 chromosomes which are coming from sperm, that is what? Male pronucleus. And both of the pronucleus will swell up and they will rush to each other and fuse with each other. And now what is this? This zygote has 46 chromosomes, 23 from ovum and 23 from the sperm and diploid number of the is restored or re-established. Is that right? Now, at the time of fertilization, when sperm fertilizes ovum, three things happen. Number one, first, second meiotic division completed. I'm repeating it. Second meiotic division completed at fertilization as soon as sperm touch. Second, another polar body thrown out. Is that right? Clear? And secondary ovum is now converted into definitive ovum or mature ovum or fertilized ovum. Right? Secondly, of course, the product of fertilization is zygote. Second thing which occurs at fertilization is that if it has brought X chromosome, then it will be female. But if it has brought Y chromosome, then it will be genetically male. So, you can say genetic sex is determined at fertilization. Third thing, sperm brings some magic molecules. We are still looking what they are. But those magic molecules activate the metabolic machinery of the ovum so that it becomes fully metabolically active zygote. How long this will live now? If it successfully develops, it may become like a human being like you, and it may live 70, 80 years. Look at the value of sperm here. Ovum without sperm never completes its second meiotic division. Disappointed, life only 24 to 48 hours and die. But as soon as sperm magic touch, rapidly completes, happily completes its first, second meiotic division, convert into definitive ovum by releasing one polar body. Is that right? And then it is metabolically activated, and then of course it will later on in embryology will discuss the zygote will go into 2-cell stage, 4-cell stage, 8-cell stage, morula, blastocyst, bilaminar disc, trilaminar disc, and then eventually embryo develop, fetus develop, baby is born, and one day it comes to Ross Medical University and starts studying it. Is that right? Any question up to this? Okay, class dismissed.